I am pleased to talk to you today about emerging infectious diseases, and that's what SARS and bird flu are. And these are primarily viruses or pathogens, often viruses, that come in general from animals into humans or into domestic animals and livestock and so on. And we see them as spreading epidemics in which nobody has ever had that infection before. And that's the case for SARS, severe acute respiratory syndrome, which you've all lived through and remember the hype in the newspapers. And the problem was, here's a virus which had never been in the human population before, was spreading seriously. And how quickly could you find a drug to contain it? Well, it's that urgency to say, how can you anticipate that more new drugs, more new emerging diseases will be found, and fast drugs will be required to control them. We were lucky with SARS. We dodged that bullet, and the epidemic was stopped before there were drugs or vaccines by good medical practice. Bird flu is another one, and that is one which is infecting lots of birds. When it infects people, terrible fatality rate. It's very different from normal flu. And so our question, Bob's and mine, was can we look at these viruses, and particularly the proteins of the virus, and see if there's anything in common which we can use to develop a strategy for control. The first thing you ever know about a pathogen these days is the full genome sequence. And you can look at the genome sequence and you can read from it where are the proteins. And we know what the viruses look like, and we can tell which proteins might be an interesting target for our study. So I just want to point out the incredible value, which you've seen in the other studies here today too, of collaborations. So I know about viruses, and in particular I've studied coronaviruses like the one that causes SARS for more than 25 years. As one of the reporters said to me when he asked me that question, he said, why? Why would anybody study it before SARS? Well, thank goodness we had done so, because then everything happened more quickly with SARS. So that's the value of basic research. And Bob Hodges, my friend here and collaborator on a program project grant, is an expert on proteins. And so we got together when this emergency came up and said, can we think of something to do? And so SARS, this is a picture of people in Hong Kong. Everybody wore these masks, which probably didn't do much good, but made them feel better. And this epidemic began in the fall of 2002 and spread starting in about February, March of 2003 from southern China into Hong Kong and rapidly spread. There were explosive epidemics, particularly in hospitals, until it spread to 30 countries, and it had about a 10% fatality rate, but a very tiny number of infected people overall. This could have been, had it become established in the human population, totally devastating. We don't have any other pathogen with a fatality rate like this in our common human pathogens. So that's why these emerging diseases are really serious. And so this is a respiratory disease, and the other target that we're looking at now is the bird flu. It's also called H5N1, and it is tremendously virulent in birds. So here's a chicken looking kind of depressed, and the red parts are blue because he's not getting any oxygen. He has a severe respiratory disease. And this can affect, if you can see all these dead chickens here, a whole chicken coop of 10,000 birds in one night. And so it's that devastating. The fatality rate in birds is just dreadful. In humans, it's also dreadful. Look at this. But fortunately, so far, very few humans can get this. So there's an incompatibility between this virus infecting birds and infecting humans. But because of this fatality rate, we need a drug. And the worry, which you've also seen in the newspapers, is when will it spread in humans the way it has spread in birds? or will it? I'm hoping it isn't going to, as everybody else. But a lot of people are developing many different strategies for quick responses. So here are the viruses. So we were trying to think, what is a common possible therapeutic target? Influenza looks like this as well. This is the virus particle. Inside is the RNA in a helical structure. And there's a membrane around here, derived by budding from the cell membranes. And sticking out from that are these spike proteins. And bird flu kind of looks the same, but these viruses are functionally totally different. Bird flu has a segmented genome, 8 different little chromosomes, if you will, and they are complementary to the message of that would be making proteins. And this is a positive sense virus. So they're really totally different families, though they look quite similar. But what's the common feature that we focused on? And that is the spike proteins. And these spike proteins are really what's on the outside of the virus that interacts with the rest of your body. Okay? So the spikes attach to receptors on the host cell surface and allow entry of the virus. And the entry of these envelope viruses is fusion of the viral envelope with the host cell membrane to dump this genetic material of the virus into the host cell and start the whole infection process. So we looked at those and discovered from looking at the genome sequence really alone that they have a similar structure in their fusion protein. And so this is a characteristic viral fusion protein. Here's a receptor binding domain up at the top of this trimer of, these can be like 200,000 molecular weight apiece, enormous glycoproteins. And they form trimers on the virus particle. And so this is the receptor binding domain. And then it is attached to a fusion domain. And this is what does the machinery to fuse this viral membrane with host cell membranes. So they're very big, they bind to specific receptors, and binding to the receptor makes this undergo a huge conformational change that leads to virus entry. And as you may guess, this whole molecule is the target for neutralizing antibodies. Neutralizing antibodies are generally ones that prevent receptors from binding. But the virus is very clever, and the virus puts out decoys. So the receptor binding site is usually in a little pocket, and around it are parts of the protein that can change its amino acids and therefore antigenicity. And they can still use the same receptor, but if you've given me a virus mutant that I've never seen before, I won't have antibody that blocks entry to the receptor site. So the receptor site itself is usually hidden in a pocket, and these are decoy sites around the top. And that means that for many viruses like flu, which you know, hepatitis C, HIV in particular, the virus is always changing those little decoy antigens around the surface, even though it keeps using the same receptor. So we wanted to find something that was not so variable, that was a common major target. And we focused on this region, and in particular, this little region right here, which I'll show you in the molecular structure here. So if you think of the spike continuing up here to the top and the membrane being down here, this is what that part of the trimer looks like. It's 3 alpha helixes. And these are rather loosely bound together in the base of the spike. And they have, you can look at the sequence and actually predict where these alpha helixes are going to be, thanks to some of Bob Hodges' programs. And so if you look at this now from the top down this way, it would look like this. And these helixes are kind of forming a coiled coil that winds around itself. And you can predict from knowing the amino acid sequence which you get from the genome, which of these amino acids here are going to be exposed on the surface, and which ones right next door are going to be buried right inside. Okay? And so the ones that are holding it together very tightly or very loosely will be the ones that are on the inside, cross-reacting across this way. The ones that are on the outside here will be antigenic because they can be detected by your immune system. Okay? And so our strategy was to see if we could develop antibodies that would stabilize this trimer. Because I told you this is a fusion machine, this is part of the fusion machine, and the first step that's necessary for the conformation changes in the spike is the separation of these three strands in this coil to coil. You have to separate and move around to start that fusion process. So what we wanted to do is to develop antibodies, an immunogen that would raise antibodies to freeze it right in that site. And this is interesting because this machinery doesn't change that much antigenically from one virus to another. And yet it's not a huge component of the virus, and it doesn't immunize efficiently because those are always coming and going, because I told you they're rather weakly held together. So sometimes they're linear and sometimes they're not. And so we wanted to say, can we stabilize this and use it as an immunogen to make antibodies that will freeze the spike in a condition where it can't freeze. So Bob developed a technique to chemically stabilize the trimer of helixes. And what he did was to take those amino acids that are on the inside of the coil to coil and make them into isoleucines and leucines, which are very hydrophobic and stick together very nicely. So that stabilized the trimer of helices. And this can all be done with peptide chemistry. I forgot to mention Bob's major thing that he does here, which is wonderful. And so he didn't use the whole spike. He just used those peptides. And he created them not from genes, but by building them amino acid by amino acid on an artificial peptide synthesizing machine. So he can substitute any residues that he wants. So what he did was to take this very stable central amino acids that made a stable coil and put our SARS markers on the outside, which would keep the antigens, the antigenic amino acids on the outside where they would be exposed only in the coil-to-coil configuration. Because if it unwinds, those amino acids will be quite separate from each other and they won't be seen by the antibody. So we're looking for conformation-dependent antibodies. And so then we use those to raise antibodies in rabbits to the amino acids on the surface, hoping that they would lock the helix and the coil in configuration. And then we tested these for the ability to inhibit infectious SARS virus infection in cell cultures. And I'll show you an example of that. And after we see if something works in this way, then we go on to see if it will also work in vivo in two different ways. So here's our spike. Here's where the membrane would be. And this is the coil that I just showed you, the heptad repeat seed coil. And when the conformation changes due to receptor binding and low pH, this has to flip up and bind onto this upper coil to coil called HRN. And you get something that's called a 6-helix bundle that draws the host cell membrane and the virus envelope membrane together. And it actually looks like this has been crystallized. So what we wanted to do is have our antibodies stabilize the trimeric coil to coil so that this process can't happen. And then virus entry couldn't happen. And so here is some data which we were thrilled to get with SARS. And I'll just walk you through this slide. Here we've used increasing amounts of this rabbit antibody, and antibodies to the upper heptad repeat had no effect. Antibodies to the lower heptad repeat, where I circled it, were very good at reducing infectivity of the virus. So if this is 100% of the infectious dose we start with, we mix it with the antibody from the rabbit or control antibodies, and then we test how much infectivity is left. Okay? These are the numbers we like to see down here, like almost 0 at 5 milligrams per 0.5 milligrams per mil, which is still a lot. And both of these antibodies from different rabbits actually stabilized that and prevented entry. So the next stage, which is in process with SARS right now, is to treat mice that are transgenic and have the human receptor for SARS, so they can be infected with SARS virus. Take those mice, treat them with the rabbit antibody to the little heptad repeat, and then challenge them with infectious SARS virus. You may imagine that this is kind of complicated because the only place the infectious SARS virus exists is in laboratories. And we have it here in our biosafety level 3 lab. We could do the tissue culture experiments like these, but we have to work with colleagues who have animal biosafety level 3 laboratories, either in Galveston or Albany, New York, to do the studies in mice with the infectious SARS virus. So there are big logistic problems in trying to work with these hot viruses. But point is, this is very promising data coming from an idea between scientists who have different backgrounds but talk easily to each other. It's always nice to have friends. And then you see very promising results like this. So the SARS stuff is moving forward, and now we're doing the same thing under the tech transfer grant with the H5 webinar. We're doing it with a mouse-adapted influenza first. And we are at the stage of having made the peptides, stabilized them, immunizing bunnies. We're about to get the antibody, which we will then test in vitro against not only the influenza virus that we're going to test in mice, but also against many other strains of influenza. You know how variable influenza can be. This is one of the most stable parts of the molecule. So our strategy is, we talk about strategies, okay, our dream is that if we can target this rather conserved part of the molecule, the spike molecule, it will not be strain specific. Does that mean we could use just that to immunize people against flu? Heaven forbid. We should use everything we possibly can. But it might be that we could use such an antibody in 2 different ways. And one way would be just like with this rabbit antibody. If somebody's terribly ill, give them the antibody passively, that they don't have to make it themselves. There isn't time. Flu is a very fast disease, unlike SARS. And it happens within a couple of days, and people are going to get sick, die really quickly. So maybe there isn't time at that point to immunize them. You could give them passive antibody and perhaps reduce the virus level, reduce the number of infected cells, and save them. Or the other, and potentially more interesting thing, is that these peptides, such stabilized peptides as Bob has designed, can be made in boxcar quantities. And that's what Amgen does up at Boulder, right? And it might be that they could be added to this year's influenza vaccine. And so before infection occurs, you might give people an immunogen that would stimulate responses against the 3 current strains of influenza, but also give them this extra one, which is cross-reactive to a site that normally isn't too immunogenic, but if you get it, it's immunogenic, and if it works, you'll stabilize all of them. So that is our plan, and that's where we are with our project. And I'd like to point out Hodges Lab, and this all requires a lot of steps. So Yanzi and Brian Trippett have been involved in making the peptides and characterizing them, and in SARS showed clearly that these antibody to the stabilized trimer only saw the conformation stabilized trimer and not the one, the spread out proteins. And Brooke Hirsch is the one who's been doing this with the influenza and is, is just about to get her antibodies from the bunny. And Scott Jeffers worked in my lab with the infectious SARS virus, in the BSL-3 lab, and did those experiments that you just saw on the antibodies made by these guys. And Xiaoyu Chen is the one who is now working on the influenza project. And the beauty of that is that we can go here directly with the mouse-adapted virus from tissue culture to the animal and prove principle if that will work. So, any questions?